my journey here today began in an HFSP letter of intent review. And sitting in that room, as he always is, was our Secretary General, Pavel Kabat. And now usually Pavel is rather circumspect. Problem? Which? What? Oh, I see. Gotcha. So normally Pavel is rather circumspect during these discussions, but he lit up when somebody mentioned the idea of using imperfect models as starting points to describe some biological phenomena. And what he said was, well, of course, if you can figure out what independent variables you need and the boundary conditions of the problem you need to solve, in other words, a sort of physical interpretation or a physical framework for solving a problem, then you can solve the problem. And I realized that he had basically simply had enunciated an issue that I was dealing with at the time. And in the process, I explained what I was doing that I'm going to describe today. And he introduced me to a number of opportunities that have allowed us to advance this story. So, in a very simple problem, there's the... Which one is this? What's going on? On my computer? What? You mean I have to do it on my computer?
Yes.
Oh, all right. Fine, okay. You have the problem that you have a bunch of objects, and these objects could be genes, and the question you have is how do I go from these collection of objects to what we call the phenotype, what manifests as a living thing. And again, if you have some coordinate system that you could describe, you could do that. And so in this particular case, it's a very simple coordinate system that leads you to this, gestalt that describes what you want to describe. But what I'm going to do today is starting off with just the general problem. The general problem is the notion of virtual cells and digital twins. Now, there's a zillion people in the world that are working on these concepts, but just as an example, the sort of general idea is that someday a doctor is going to be able to take a sample from you, a biopsy or blood or whatever, and there's going to be a fingerprint that, with some overarching model, will predict or diagnose exactly what your state of health is or what disease you may have. But that starts with a virtual cell. And the question is not only what is a virtual cell, but how do we get between the virtual cell and the digital twin? So going to the virtual cell, generally what's been driving all this mania has been the transcriptome revolution, the fact that people can do single-cell analysis of entire transcriptomes and therefore be able to describe with a molecular signature each and every cell in a tissue or in a pathological state versus a normal state or during development or whatever you could possibly imagine. But again, and this particular slide happens to be borrowed from my friend Sarah Teichmann, who's in charge of the Human Atlas Project, a very large program looking at virtual cell and virtual tissue development. So I want to acknowledge all the people that were involved in this before I forget in the end. And so the most important people have been my young colleague, Adrian Cerrillo, this very, very talented PhD student, Jacob Bolak, and Xavier Giroir, who's just a master's student in my lab, is now doing his PhD at the University of Cambridge. So there's 4 parts to this discussion. What I'm going to do is first try to convince you that these models of the cell, molecular representations of models of cells, can be described at different levels of hierarchy of information. And I want to convince you that the most important level, and the one that we have to access in order to come up with truly physical models of the cell, are at the level of protein-protein interactions. And then secondly, I'm going to explain a rather remarkable way that we can develop a thermodynamic model of PPI networks, not simply topology, but actually the thermodynamic information that goes into that topology. And then I'm going to give you a couple of applications of this to an actual problem of cell identity. And so, starting with this, if we start at the level of the gene, okay, at the bottom, at the bottom, what you can see is, what you could say is, okay, we know that there's variations in the genome, there's variations in the environment, and we want to map those to different levels. So the easiest one is at the level of mRNA, but the problem with mRNA is that all you have is the information about what a single trajectory, not the relationships between the elements of those trajectories. In other words, the relationships between the genes. You might be able to extract that information, and there are methods for doing that, but if you can go to the levels of proteins and protein-protein interactions, now you have a physical signature of how genetic variation and environment affect an organism, and therefore you're as close to phenotype in a molecular sense as you possibly can get. And that's because basically protein-protein interactions capture the entire fate of a gene, really. Everything that passively happens to the proteins and their interactions, post-translational modifications, protein and RNA homeostasis, all of that is captured finally in protein-protein interactions. And this is just a different way of representing that. But what this represents is the fact is that when you perturb the cell or an organism, either in the environment or in the genome, then where you're seeing perturbations in the protein-protein interactions are mapping you right to the physical consequences of those perturbations. And here's an example of a paper that is about to be published from our group. Just look at the left-hand representation. What we've done here is we took strains of the yeast S. cerevisiae with different genetic variation, and what we did was we probed protein-protein interactions in living cells, and we asked how are they being perturbed relative to perturbations in the genome. And these are the results here. And the most important, on the x-axis is the prevalence, and on the y-axis is the size of the effect of genetic variations in the form of single nucleotide polymorphisms. The most extreme is the blue one, okay, and this is simply the open reading frames. And that's obvious. Variations in the open reading frames are likely going to give you different phenotypes. And we see that at the level of protein-protein interactions. But unlike with when you probe RNA, we're picking up all the consequences of coding and non-coding, and those are the other curves you see there that include both 3' UTR effects and non-coding RNA effects. So, and that's the point. Now, this idea now applied to virtual cells has not been lost on people in the field who've taken transcriptome data and then in sort of generative neural network models have embedded on top of that protein-protein interaction networks inferred from transcripts. Now that's fine, except all you have is the topological information. There's no thermodynamic information. So a clue to a remarkable phenomenon came up again in this study of protein-protein interaction effects on genetic variation. What this result here shows is Pearson correlation coefficients and frequencies in a set of 48 of these strains that we used in this protein-protein interaction analysis where we did two things. We either looked at random pairwise correlations between RNA across these different strains as measured by RNA-seq, or pairwise RNAs coding for proteins that interact with each other. So you can probably already guess which distribution is which. The gray distribution is the one for random, and the blue distribution is the one for protein-protein interaction pairs. Now think about this for a minute. What this is saying is that somehow protein-protein interactions are talking to the RNA. And how they're doing that, we can discuss at the end, but the fact is it's happening. But then you can make a second inference, which is that if this is true, then you can do the reverse. Maybe somehow from the RNA information, you can go to protein-protein interactions. And just quickly, I'm going to skip over this because we're running short on time. Okay. So the first thing is that we needed, in order to be able to do this, if we could make any predictions, we would need some sort of ground state truth. And we were able to get this from a series of data from Matthias Mann's lab that described both the interaction stoichiometry from affinity purification mass spec studies and abundance stoichiometries. And I'm going to go very quickly now. From these studies, you see interesting sorts of relationships. In other words, you see cases like this, where in this particular study, it was Cho et al., they showed that the interaction stoichiometries correlated with localization similarity. And in a commentary that Emmanuel Levy, who's in the room with me today, wrote, what we realized was that what this describes then is that you've got a bunch of you actually have a huge number of lower stoichiometry interactions in the background of a bunch of very strong interactions or modular interactions. And carrying this further, what this means is that on this axis, from binding stoichiometry, you can obtain PPI, you can obtain binding constants, dissociation constants, KD, and you can obtain binding specificity. And now this becomes this coordinate system that you need to define the molecular organization of a cell. And in fact, it does. And without going into detail, all of these different characteristics here, including genetic interactions, correlate with KD. Okay, so we're able to calculate KDs from these data, and we used a very simple random forest model containing a set of additional features, including mRNA levels at protein localization, gene expression, gene co-expression, and string scores. And these are the results we got for yeast, for both fraction bound and for binding affinity of prediction versus experiment. And these are the results for human. This is a summary. And for both yeast and human, and you can see the limit of experimental reproducibility was that for affinity purification mass spec experiments. And so in an application of this, we can show how does this look compared to, how do our results when we do a projection of either functional binding or KD onto the transcriptomes of these different cell lines, how does that compare to, say, the best method that exists today, which is SCNet? And as you can see, it performs pretty well, just as well. It also, when we look at enrichments for specific cell processes, this is for CD4 positive cells, we get the same patterns as for our results, which are on the left versus SCNet on the right. But we get different distributions of where we see enrichment for this particular cell type, the CD4 positive helpers. So finally, the question is, what is the origins of this identity that we're seeing? The question is it these modular tight interactions with high stoichiometry, or is it this vast sea of connectors? And the short answer, I'll tell you, is that the identity is captured in the connectors, not in these modules. And you can see this also within data, data derived from the string compendium, which is a collection of information and scoring function for protein or gene associations. And what I show here is that the sort of high scores that people usually use to define interactions are bent towards the high affinity, which is the higher scores there in orange, and the blue scores are not as represent sort of poor string scores. In fact, my argument would be, based on our analysis, all of string is good. You shouldn't use any sort of thresholds with these things. So I think I'm going to stop there because this was much more information than I'd hoped for. And if you have any questions, I'd be happy to entertain them. Thank you.
So we have time for a few questions. Yeah, please.
So I don't know if I got it wrong, but so my understanding is that you were able to transform transcriptomic data into interaction data, and then this resolves much better the differences between cell types. Now, do you need to have the interaction data for the cell types before? Like, do you expect other cell types that we don't have this interaction data for to, we could use the same methods? Or?
No, our model is trained on an individual cell type, and then everything else comes from the RNA and the string scores. And I can't, for those of you who are aware of string and the utilization of string, I can't stress more how important it is. And it's remarkable that this contribution of Per Bork to the field was basically, without knowing anything about protein thermodynamics, his string scores are a major contribution to our model.
Hi.
Yeah, please. Go ahead.
Hi, thanks. So this makes me think of gene regulatory networks, and I don't know if it's a correct analogy, but if this makes any sense, would you then have something like in gene regulatory network theory, there is a concept of kernels that are some key, let's say, modules that define some identity of cell types. So do you think you could also have some key modules that are really signatures for cell types, or is the global?
I think it's exactly the opposite, in fact. As I was saying, it seems to be that the connectors are what are driving the distinctions between the different states. And I should stress, I mean, let me go back, because I went over this way too quickly and it's a complicated figure. Oh, don't go that way, that's a mistake. Okay, here. So what we're looking at is these plots are for different cell types, and on the x-axis are differences in fraction bound between one cell type and all the rest, and on the other axis is KD. Okay, the higher the KD, the higher the number here, the tighter the binding, the more likely that these are modules. And what I'm saying is that the differences seem to be clustered in the lower affinity interactions, which are not modules. They're connectors. They connect the modules together. And that seems to be what's defining identity.
Hi, over here. You showed this mRNA abundance plot for random pairing and for the pairing of the interacting proteins.
Yeah.
And that's obviously quite interesting, right? Now the question is, you said that shows that there's some feedback between the interaction and the mRNA level, but couldn't it just be a genetic adaptation that, of course, the expression levels are a priori coordinated by evolution, that they are in a similar range if the two proteins are interacting? Like, how do you know that there's a live feedback going on?
Well, I don't have anything to represent this here today, but what we're seeing is that these numbers change under different environmental conditions. And that's saying that something has to be happening at the translational level, I think. There is some theory, a woman named Christine Meyer has been doing has some interesting ideas about co-expression of proteins that form complexes, and that would be consistent with this idea. Well, no, Christine does, and this is essentially what she says. Okay, this is one version of a model that she's proposed. If you're interested, please read this commentary here. But the basic idea is that you have two subunits shown here in light and dark blue of a protein complex, and somehow one of the subunits that's already been translated is associating with the RNA of the other partner, and somehow controlling, say, it's either the rate of translation or something to control the stoichiometry between the two complexes. So you can imagine something like this. If you added onto that any disequilibrium of the stoichiometry led to degradation or inhibition of degradation of the RNA, that might explain why these numbers correlate with the numbers of the concentrations of RNA correlate with association of the proteins.